Now, on to today's program. Continuous manufacturing in biologics has proven a very ambitious endeavor, while measurable progress is being made on automating upstream operations. We're a long way from the coveted lights-out manufacturing paradigm, specifically on the downstream side. But technologies that support the ideal are rapidly improving, including the process analytical technologies that are foundational to the effort. On today's panel, we're going to address the state of continuous manufacturing in biopharma, what's standing in the way of continuous becoming the standard, and what technologies such as PAT, biopharmaceutical organizations can be adopting to build the foundation for continuous tomorrow. Joining me for the discussion are Barrett Fallentine, Director of Process and Product Development at Pharmatech Associates, and Paul Testa, EVP of Supply Chain and Operations at Kyowa, Kirin, North America. Process development has been Barrett's bailiwick since he began his career in the late 1990s as a PD scientist at Bayer. He subsequently held key process development positions with Corixa and Coulter Pharmaceuticals, Medarex, 5' Therapeutics, and Coherus Biosciences before joining Pharmatech Associates in 2017, I believe, to share his experience with the broader biopharma community. For his part, Paul brought 10 years of technical operations experience at J&J and 8 more years at Shire to Kyowa Kirin when he joined the company in 2019. KKNA is deploying proprietary technologies to the development of next-generation antibodies in nephrology, oncology, immunology, and allergy, and neurology. Unfortunately, flagship pioneering medicines, Dr. Palani Palaniappan, who is scheduled to join us this morning, will be unable to make it due to an unexpected snag and some international travel, meaning the airline rescheduled his flight several times over the course of several days, and he is presently somewhere over the Pacific. So, Paul and Barrett, welcome to the trio, as it were, and thank you for joining me.
Thanks, Matt. Happy to be here.
Likewise. Thanks, Matt.
I'm happy to have you guys here. I want to roll right into the conversation here. And Barrett, I want to get started with you because, as I noted, the position that you took there at Pharmatech exposes you to the process development operations at several biopharmas. So I want to start with sort of a level set on where we are. As you survey, Barrett, the landscape from your purview, what is the state of continuous manufacturing biopharma today? And I'm going to make it a two-part question to start with. Who's adopted what and to what end?
Well, Matt, I think you touched on it perfectly in your intro. I think right now there is no, quote unquote, true continuous in biopharma yet. I think we have somewhat of a hybrid system. As you mentioned before, upstream operations, there's been a lot of advancements going on with continuous. I mean, I started my career at Bayer and we were doing perfusion, and that was in the 90s. That has only improved since then. The downstream operations, that's a different story. I think, with single-use technology and the adaptation of, kind of QBD principles, I think we're getting there. I have heard that, some of the industry giants, possibly Sanofi, I know they've cobbled together a complete downstream system. And again, I've heard this, I haven't seen this, but I think that's kind of where we are. I think there's not a ton of expertise as far as having continuous like there is in solid oral dose. And I think that's one of the challenges. And there's also significant capital investment as well. So I think that's another kind of roadblock that we have to overcome. But anyway, I think that's kind of where we are right at the moment. But things are still kind of evolving.
Yeah. What do you see standing in the way? You mentioned, I heard you use the words cobbled together, so that to me, it sort of indicates strong desire, perhaps desire that's coming a little bit early in terms of the actual solutions available. So I'm just curious from a motivational standpoint, what's driving it and what's standing in the way?
Well, I think motivation-wise, again, just kind of like other continuous processes, you're going to get, if you go full continuous, you're going to get a higher quality product. if you're doing multiple sampling points and monitoring real time that has real time feedback, it's only going to improve the quality. I think that's the main motivation for, you know, folks in the biologics field to try and implement this from a kind of a soup to nuts point of view, just to improve product quality. But I do think, you know, again, also like solid oral dose, like there are roadblocks. I mean, I think expertise is one. I think this is relatively new. You know, when you're talking about downstream operations, you have multiple sampling points. There's multiple hold steps that you're going to have to overcome. If you're regenerating columns, you're going to have to figure that out. So I think there's, I mean, academically, it's kind of an academic exercise at the moment, and people are definitely doing it, but it's not completely, fully a true soup to nuts end-to-end process yet. Mm. And I think that's where, you know, these PAT technologies can only help, you know, alleviate some of those bottlenecks.
Yeah, and we'll get into some specifics there in just a second. But Barrett, one more question for you specifically before we roll on. As you survey the your client base in bio specifically, how would you characterize or describe the appetite, the drive to move towards continuous?
Oh, there is a very large appetite. I see this. You know, I came from kind of biosimilars prior to Pharmatech, and I know when you're looking at biosimilars, generics, what have you, there's a huge appetite for it just because there's a huge market for it. So I think there's definitely room to grow and room to advance continuous. I definitely think there's a huge appetite for it. It's just a matter of logistically implementing it.
Yeah. Paul, I want to turn to you, and I'll frame that question up sort of specific to KKNA. What's the appetite at KKNA to move toward continuous in bio specifically? Because we're going to talk a little bit about the fact that you guys have a more diverse portfolio with some small molecule as well that might look a little bit different from your antibody endeavors. But what's the appetite on the antibody side and the other biologics KKNA is working on to move towards continuous?
Yeah, I mean, great question. So in the same way that Barrett just described, I would say we've got a good appetite to move in that direction where it makes sense, right? I think the biggest obstacle, so Barrett talked a great deal about, some of the science and some of the application challenges, but, the finances are not, too far down the list from an obstacle perspective. So for us, where it makes sense, where there's a product that is already, certain to be a success from a commercialization perspective, okay, fine, let's go forward. But that needs to be justified, not just financially, but certainly from a quality product perspective, from an economic perspective in terms of the payback, right? And that's where I think one of the bigger challenges would be. So to answer your direct question, for us specifically, yeah, we're interested. We're interested where it makes sense, not just as an academic or scientific exercise, but from a business application perspective, as it relates to regulatory filing, the success of the data, the quality of the product, the consistency of the therapy as it relates to the patients. All those things are important to us. But of course, in the biotech world, sometimes the patient population is smaller, so therefore that financial justification becomes a little more challenging. Of course, if you've got a multi-billion-dollar blockbuster, all right, maybe that's a different equation, but you don't actually know that in phase one, which is where a lot of that activity really needs to start to take place to drive the process automation and the critical to quality attributes and the return process parameters, such that you can then make the adjustments in process. So it all kind of fits together like a jigsaw puzzle for me, and it's an important element to be strategically consistent in terms of what you're trying to accomplish, but also why you're trying to accomplish those elements.